Unit 2 starts with Chapter 6, which is about another group of microorganisms, the viruses. Virus general structure can be either considered naked or enveloped. So first we'll discuss the naked viruses, which in the interior contain a nucleic acid that can be either DNA or RNA. Surrounding the nucleic acid is a layer called the capsid, which is made up of smaller subunits called capsomeres, and the capsid is made of protein. Outside of the capsid are spikes, which are protrusions from the capsid that will be very important in infecting host cells. And I want to remind you that the viruses are acellular microorganisms, so although the capsid structure resembles a cell in some ways, it is not considered a cell. So the virus is acellular. The envelope virus, also acellular, and looks very similar to the naked virus with the addition of an extra layer called the viral envelope. And the viral envelope is made up of lipids. So looking at this with text, we have the covering will be capsid in all cases and an envelope in the enveloped virus, not found in the naked viruses. The central core or the components of the interior are going to be a nucleic acid, either DNA or RNA molecule, and occasionally we'll find proteins, specifically enzymes that will perform functions that the host cell cannot do for the virus. The genome of the virus, as I mentioned, can be DNA or RNA, and that DNA and RNA can be either single-stranded or double-stranded. So typically in cells, we see double-stranded DNA and single-stranded RNA, but in viruses, we can see combinations in all forms. The RNA viruses will often have an enzyme called reverse transcriptase, which can convert DNA from RNA. With respect to size, viruses are the smallest of all microorganisms. Shown in this figure from your textbook, virus structures are much smaller than bacteria cells, which are in turn much smaller than the fungal yeast cells shown in this figure, and also much smaller than human cells. So overall, the size of virus is very, very small. And for this reason, viruses are filterable. So if you take a liquid that contains microorganisms and you pass it through a filter, which has very small openings or pores, bacteria and other larger microorganisms will not pass through the filter, but viruses, because of their small size, will pass through the filter. So you cannot sterilize virus-containing liquids with a filter because viruses are filterable. Also, viruses are technically not considered alive and are often referred to as biological entities. In Chapter 1, viruses did not fit into the phylogenetic tree of life because they technically are not living. So this slide lists reasons why viruses resemble life, but the number of reasons why they are not considered alive outweigh the number of reasons why they are. So generally, viruses are not considered alive. Viruses are obligate intracellular parasites, which means they are inactive outside of a host cell. They have to get into a host cell in order to reproduce, and so they live intracellularly, and they are obligated to be intracellular because they cannot reproduce without being inside of a host cell. The word parasite here implies that the virus being inside the host cell is harmful to the host cell. So the virus's relationship with the host cell is a parasitic one. We'll first start discussing animal viruses and the multiplication cycle that they go through. Notice we don't use the phrase life cycle because viruses are not living. So the animal viruses are viruses that infect humans and any other animal. And the first step in the multiplication cycle is the adsorption step. So in this step, the virus comes into contact with its host cell, and the contact between the virus is made with the host cell because the viral spikes are going to connect with a receptor on the surface of the host cell, which enables the virus to adhere or stick to the surface of the host cell. The spike receptor interaction is very specific, and for this reason, viruses can only infect certain types of host cells. The next step in the multiplication cycle of animal viruses is the penetration step, where the virus gets into the interior of the host cell. In figure 613, we see in part A one way of penetrating by viruses. The virus penetrates the host cell in part A by endocytosis, where the contents and the entire virus are brought into the host cell in a vesicle. Then the vesicle is broken down. The capsid and perhaps the envelope are also broken down, and then the nucleic acid will be free inside the host cell at the end of the uncoating step. In fusion, viruses penetrate the host cell by the envelope fusing with the membrane of the host cell so that the contents of the virus can get into the interior of the host cell. Then the capsid is broken down in uncoating, and the viral nucleic acid is free in the host cell interior. Naked and enveloped viruses can penetrate a host cell by endocytosis, and enveloped viruses can penetrate by fusion, and only enveloped viruses. Once the viral DNA or RNA is in the interior of the host cell's machinery is taken over. Mostly the ribosomes and enzymes of the host cell will now be used to make viral proteins and viral nucleic acid. So in the assembly stage of the life cycle, all of the components of the virus are made inside the host cell by the host cell machinery. Once the virus has been all the viral pieces have been collected together into a virus, the virus will then exit the host cell in the release step. And shown in this figure is release by budding, but release can occur by budding, which is shown on the right in this slide, or by lysis, which is shown on the left. So naked animal viruses will lyse their host cell, meaning the host cell gets completely full of virus and the virus will burst out of the host cell. The enveloped viruses will bud off, where they take a layer of the host cell membrane with them, which will become the viral envelope during the budding stage. In an animal virus infection, there are 3 outcomes. The first is cytopathic effects, which means that the host cell is damaged and its function is somehow compromised. The second outcome is a provirus and a persistent infection, where the virus can insert itself into the host cell DNA and then can exist in the host cell without doing anything for a period of time before it is reactivated and can then cause a new infection in the host. So an example of a provirus and persistent infection is the chickenpox virus can resurface many years later as shingles. The last outcome of a virus infection is oncogenic transformation, where the viral DNA inserts into the host cell DNA and can cause the host cell to become a cancer cell, which is what happens in hepatitis C. Another kind of virus we'll discuss are the bacteriophage, which are viruses that infect bacteria cells. So their difference from the animal viruses is that their host cell type is different. Where the animal virus infects human cells or animal cells, the bacteriophage infects bacteria cells. Bacteriophage are made up of a head region, which looks like a capsid containing a nucleic acid, tail fibers, and a tube. And the bacteriophage shown in this image is sitting on the surface of a bacteria cell and is injecting its nucleic acid into the bacteria cell's cytoplasm. The multiplication cycle in bacteriophage is very similar to animal virus multiplication cycle, with the addition of a lysogenic state, which we will discuss in a moment. Figure 619 shows the multiplication cycle of temperate and lytic phage. So we'll first start discussing lytic bacteriophage, which start with adsorption. Their tail fibers attach to a receptor on the surface of their bacterial host cell. In step 2, penetration, the tail fiber and tube are compressed to inject the bacterial cell with the viral nucleic acid. In step 3, 4, and 5, the components of the bacteriophage are replicated by the host cell machinery. They are assembled into small pieces, and then in maturation, those small pieces are assembled into whole viruses. In step 6, the host cell is lysed and the bacteriophage virus are released in step 7 to go infect other bacteria cells. So the lytic phage follow steps 1 through 7. Temperate phage are phage that are capable of exiting the lytic cycle to enter something called the lysogenic state. So a temperate phage will adsorb to the surface of its host cell, inject its viral DNA into the host cell cytoplasm in penetration, and then at this point can exit the lytic cycle and enter the lysogenic state, where the viral DNA becomes a prophage that is inserted into the bacterial DNA. So in the image of the cell in a lysogenic state, the blue region is viral DNA and the pink region is the bacterial DNA. So they have been combined into one piece. The reason the temperate phage enter the lysogenic state is although new viruses are not being made, each time the bacteria cell replicates DNA and divides to make daughter cells, the bacteria cell will replicate the viral DNA and pass it on to its daughter cells. So in this way, the phage can spread through a population without actually making new viruses. A virus that is a temperate phage can stay in the lysogenic state for a number of generations until it exits the lysogenic state and re-enters the lytic cycle. With steps 3, 4, and 5, the temperate phage pieces would be replicated and assembled into mature viruses. And then in step 6, the temperate phage would lyse the host cell and go on to infect other bacteria. So that's the end of chapter 6. So you have half of the material you need to do your second homework.